Hello people, this week we will study how to model excitable media using cellular automata. We define an excitable system as a system that consists of three main states. Those states are rest, excited, and refractory or recovery. If the system is at the rest state, it can become excited through a supra-threshold stimulation. From the excited state, after a while, the system transitions to the recovery state. It then takes more time that the system backs to the rest state. By the way, supra-threshold refers to a stimulus that is large enough in magnitude to produce an action in an excitable system. Now I want to introduce excitable media. An excitable medium is an excitable system that can propagate a wave until a certain amount of time has passed. We refer to this certain amount of time as the recovery time. It should be noted that during the recovery time, the system cannot propagate another wave. For instance, a forest is a good example of an excitable medium. If a fire burns through the forest, no fire can return to a burnt spot until the vegetation has gone through its recovery time and regrown. Neuron in our bodies is another example. Neurons are first excited by a stimulus and then return to the rest state after a recovery time is passed. Cardiac tissue in our heart can be also considered as an excitable medium. Cardiac tissue keeps the heart pumping blood across the body. The cardiac muscle tissue contracts once stimulated by electrical impulses, and then, after a while, return back to the rest state. In today's lecture, we are going to simulate how cardiac muscle tissue works. Cellular automata provide a simple model to simulate and understand the behavior of excitable media. Each cell of the automaton represents some section of the medium that we want to model. For example, a cell can correspond to a patch of trees in a forest, or a segment of heart tissue. In excitable media, each cell of the automaton can be in one of the three following states. State 0 represents the resting state. In this state, the cell is unexcited, but can be excited by a supra-threshold. State 2 is the excited state, and an under-recovery cell is in state 1. In this state, the cell has recently been excited and is temporarily not excitable. In excitable cellular automata, the state of a particular cell in the next time step depends on the state of its neighboring cells at the current time. As shown here, excitable cellular automata consider 4 neighbor cells of each cell to compute its next state. As mentioned last week, the new state of each cell is determined according to some fixed rules. By applying the rules to all cells in an automaton, the next generation of cell states is computed. In excitable media modeling, there are 3 rules for computing the next generation of cell states. Here I have listed these rules. Based on the first rule, if a cell is in the resting state, it remains in this state unless one or more of its 4 neighbors is excited. The next rule says that if a cell has been already excited, it transitions to state 1 at the next time step or at the next iteration. Finally, If a cell is in state 1, it transitions to state 0 at the next iteration. Simple cellular automata typically don't model well the behavior of excitable media. To obtain more accurate results, I want to introduce another model. Gerhardt and his colleagues revised the cellular automata model explained in the previous slides to create a more realistic model of the excitable media. In this approach, each cell has two internal state variables, U and V. U can have two values, 0 representing the unexcited state and 1 corresponding to the excited state. The state variable V is a real number ranging between two endpoints, 0 and Vmax. If V is 0, it means that the cell is in the resting state. Vmax represents the state when the cell is completely refractory and fully unexcitable. In this model, as we can see here, each cell has a square neighborhood of radius r. As mentioned before, the cell state is updated based on the current state of the cell and the states of its neighboring cells. It should be noted that larger neighborhoods give finer spatial resolution to our automaton. Here we have rules for computing the next generations of cellular automata. Regarding the first rule, an unexcited cell can be excited if the number of excited cells within its neighborhood exceeds a certain threshold KEXCI. The next rule says that the V variable of each excited cell is increased by G up in each iteration. G up here is an integer number. The cell transitions to the unexcited state once its V reaches the fixed value Vmax. Based on the last rule, if a cell is in the unexcited state, its V variable decreases by G down. Once V reaches near to 0, it transitions to the resting state and it is ready to be excited again. Like G up, G down is also a positive integer. In this model, as you might recognize, we can control the transition speed between the states using the state variable V. Now I explain how excitable cellular automata can be used to simulate the heart. Heart muscles are made of tiny cells. Electrical signals travel through the network of cells which stimulate upper and lower chambers to contract, creating a heartbeat. Generally speaking, the electrical signal starts in a group of cells at the top of the heart called the sinoatrial or SA node. The signal then travels down through the heart, triggering all resting cells in the upper and lower chambers. In other words, the electrical signals stimulate the cells in the heart tissue to contract. After the chambers contract, each part of the system electrically resets or recovers itself. So, based on what explained, we can conclude that the heart tissue can be considered as an excitable medium, consisting of many cells. Each cell can take one of the three states, resting, excited, and recovering. In the recovery state, the tissue slowly returns to the rest state. The cells of the SA nodes are known as the pacemaker of the heart because the rate at which these cells send out electrical signals determines the heart rate. In medicine, arrhythmias refer to abnormal rate rhythms and could cause death. So, computational models of the human heart are used to address current clinical problems in cardiac electrophysiology and open the doors for simulation-guided approaches to anti-arrhythmic drug development. We can simulate the heart operations using the Gerhardt Cellular Automata. As presented in this slide, Gerhardt Cellular Automata simulate the propagation of electrical signals as a 2D spiral wave. At the center of the wave, we have the signal generator or the pacemaker. The black cells represent cells in the excited state, and the white ones corresponds to the resting cells. In biology, normal rhythmic behavior and many abnormal arrhythmic phenomena can be studied using 2D models like this. In Lab 6, we will implement the simple and Gerhardt cellular automata and study the behavior of the spiral wave with different parameters. Here, we have one article for further reading. Okay, that's all for today's lecture. Thanks for watching this video.